in the world. And if you notice, anthrax, bioterrorism. Are there idiots in the world? Yeah. So we are in that ongoing arms race, whatever we call it, with these pathogens. All they're trying to do is replicate and go on to create the next generation. But again, so are we. They have their agenda, we have ours. So we are in that struggle. So, but the thing is that over time, what the focus is on is actually going towards coexistence. If a pathogen kills you off too quickly so you can't spread a disease, how much use is that to the pathogen? Not really. So over time, there tends to be mitigation. The bubonic plague isn't as deadly as it was 500 years ago. Ebola actually is slightly as dangerous than it was. Still don't want it, but it tends to be that mitigation. And that mitigation, that increase in mortality and morbidity will continue until it gets to this balance. Is it spreading fast enough without killing too many of us? Doesn't mean you're not going to get killed, but it means that we are actually at a balance. It's a trade-off between what's going on. It doesn't always work, because there's always these mean, nasty ones that are just the punks of the class who just want to go in and wreck everything for everyone else. So we have like a pathogen has a superior ability to kill. It just basically wipes out the host too quickly, loses nutrients, isn't able to replicate fast enough because the host dies, and therefore can't spread. It's a dead end. And we'll talk about commensality in a few seconds with that. There are certain types of, how should we put it, sharing mutual living space with pathogens. You've got predator, prey, you've got parasite, host, you've got commensality, you've got mutualism. There's a whole bunch of them. We'll cover this in a few seconds. But basically, as the pathogen becomes essentially weaker, we use up less resources trying to fight it off. So that's the benefit for us, is that we are not having to, say, have a high fever, we're not having to ramp up our immune system so much to fight it off. And many of these pathogens then end up, and they can become part of our own microbiome. So they go through something that's trying to kill us immediately to something that lives with us, and actually sometimes we cannot live without. Does that mean that they won't take advantage of the opportunity to cause infection? That's that for you, this is a good one. Lives very happily up our noses, yet it can cause furuncles, it can cause a whole bunch of stuff. It's the wrong place all the time. So we talked about exposure, infection, disease. There's another one that sneaks in, colonization. So when we talk about infection, what is that implying? There's a possibility it's going to lead to disease. What about colonization? Is that a bad thing? Lactobacillus in the vagina. Is that a bad thing? It's a good thing, because it produces acid, which then keeps down other pathogens, blah, blah, blah. So those invading organisms do not have to be pathogenic. They can be beneficial, in which case we want those colonies. Then the invading organism, if it's kept under check by our microbiome of our immune system, everything else works out. In these situations, one of the, if we have a pathogen that gets in, we have a couple of different scenarios that can happen. One, the pathogen kills us. Two, we kill the pathogen. Or three, it becomes that stalemate situation. Okay, and we can end up with that mutually beneficial relationship, that mutualism going on, where both benefit. Infection, obviously we now have a problem because that pathogen is now latching hold of, taking up real estate, and is now setting you up for disease. Remember that infection does not necessarily mean disease. Disease is when you start the symptoms. During during the infection stage, there's a fight for all the resources, but there may not be any external, or at least any manifestation of symptomatology. And then disease, well, that's when you got your fever, that's when you got your rash, that's when you got your throat up, and all that stuff. Okay? So again, 3 different outcomes. Immune system can win. Just because your immune system wins against the pathogen doesn't mean you get away scot-free. No. You don't need a lung transplant. Could be in a wheelchair for the rest of your life. You might end up with significant nerve damage. Yes, you survive, but it's not good. We end up in chronic disease because your immune system and the pathogen are trying to take each other over, and they're not quite winning either way. And then we've got death. So it's almost like endo lectures, next trimester. It's like every other slide ends in death. It gets a bit morbid after a while. Okay? Modern medicine, apart from some of those kind of hippie new age wellness doctors out there, the main focus of modern medicine is disease, because where's the money? Whoops, no, it's always going to be a problem. Let's spend millions and millions of dollars on pharmaceuticals that work half the time, if you're lucky. Again, colonization, exposure colonization does not necessarily lead to infection, and infection doesn't lead to disease. Okay? This week really is just going through a lot of terms that you're exposed to, but we should really clarify. So incidental infection, that one. Good example, the Western and Eastern equine viruses. They can get into us, but they can't get out. We're an incidental. In fact, we get incidental infections. Okay? We're not part of the normal situation. So an incidental infection is when you get infected with a disease or with a pathogen that can't get out of you. So that means that you're a dead-end host. Incidental infections happen in dead-end hosts. Okay? Doesn't mean that it's not going to try and kill us. They can't get out. Communicable diseases, that's anything that can be passed from one person to another. Non-communicable, obviously, in that way, that's from dead-end host to dead-end host. Now, these are epidemiological terms. So do they make kind of sense? Some of them are a bit weird. But yeah, if it's a non-communicable disease, well, how much of a disease is that? Well, you can't pass it on. So I can't pass on Western equine virus to like Alex, because he keeps on looking at me, he's freaking me out. Okay? But I can directly pass to him. So between you and me, it's non-communicable. But mosquitoes can pass it everywhere around. Does that make sense? So this is species dependent. Just because I can't pass it to you doesn't mean it can't be passed from other species to other species and everything else. When we talk about after effects, we could use the term consequences, but in medicine, why would you use the word consequence when you could use another silly word? Sequelae. Anyone heard of that one before? Sequelae of disease. Sequela. Okay? Someone opened the dictionary and went, oh, use that word. Yeah. So, you're talking very quickly, just 1 second. So basically, you're talking to a patient. So the sequelae of this, does that sound not too bad? Consequences of this disease. It kind of freaks people out a bit. Is there a question?
Did you say C? It starts with a C.
It's not with a C.
Yeah, that's what you said.
We didn't say C.
Oh.
Consequences starts with a C. That starts with an S. You're mental, not getting that mental, are we? Okay. Going back to the human microbiome, we probably understand about 2% of it. We do have a whole bunch of potentially pathogenic microbes all over us, but they kept in check all the time. Isn't that a wonderful thing? That's why we don't have staph infections going crazy. But as soon as dysbiosis occurs, then all bets are off. We do have certain tissues that don't have any microbiome whatsoever. Strange enough, your blood. Do you want your blood to have some pure and clean? cerebrospinal fluid, synovial fluid, liver, kidney, all those different things, brain. They are considered sterile, which means if a pathogen gets into them, they have no microbiome to help defend them.
Does dysbiosis often result when organs are perforated, like in a gunshot, which like exposes those pathogens to other
I
environments?
would say gunshot is an extreme version of a dysbiosis. Yeah.
Yeah.
If you shoot someone in the spleen, what happens to part of their immune system? Yes. If you rupture the spleen, then yeah, so you have a significant impact on immune function, therefore you're immediately in the dysbiosis. Good point. So all you have really in that situation is our immune system, but the microbiome and our immune system keeps that going. So these are some of those terms I mentioned earlier on. Mutualism. If something's mutual, I get something and you get something. Does that mean we get the same amount? No. But it has to be a reasonable amount to make it worth my while. If I have a teenager living in my basement, I kind of expect them to take the trash out from time to time and maybe do their own dishes. And then I'll feed them pizza and hope eventually they'll get a job and move out. Parasitic. Host organism suffers while the invading organism benefits, and that was used on these diseases. There's plenty of examples of that. You don't have to be a parasite, as in microbiology terms of a parasite, to be involved in a parasitic action. Metabolism, as we already talked about, one gains something, the other gains nothing. If one gains something and the other loses something, that's parasitism. But with commensalism, it's a wash. You don't even notice it's there. Something's gaining, but you're not losing or gaining anything. And this is one thing we need to take into consideration, depending where it is. These microbes can be in one part of your body involved in a parasitic relationship, another part of your body, mutualism, another part, commensalism. It all depends where they are and what they're doing. Okay? Just because you might have, I don't know, a bit of folliculitis going on your skin, does that mean that it's going to be everywhere? No, because the rest of your body is in a state of essentially symbiosis with everything else. Medications, temperature, hygiene, all these different things will affect this. That diagram shows some of the variations. Your intestines, you've got about roughly, there are 1,000 different species of bacteria. Just in your gut. 1,000. Okay? Oral cavity, 300 to 500. So people, the scientists who got out and said that the mouth of a pig is actually cleaner than the mouth of a human. So it depends on the human.
Depends on the pig.
What's that, sorry?
It depends on the pig.
No, I've known some very clean pigs. Okay. Okay, on the side, on one occasion, I had some friends in the countryside in England that were raising a pig. It was a Tamworth, beautiful orangey brown, awesome, and it would follow them around all over the place. I'd go over, pet it, awesome. And then a couple of months later, I go back over and I asked how the pig was doing, and they said, "How does it taste?" Because I was chomping into the food. It's like, I'm going to keep, to honour this pig, I'm going to keep it eating because it tasted really good. Anyway, your human microbiome is not built in a day. Starts off when you're first exposed. What's the first exposure? Well, hopefully birth. When it's in your womb, that's the pathogens getting in. So you, in theory, want to be in a sterile environment within the womb. Human fetus in a sterile environment. As soon as that mucus plug starts breaking, the water barrier, the waters break, now there's a potential for pathogens to get in and cause problems. It's also impacted by if the baby is delivered C-section versus vaginally. If it's delivered vaginally, then it is exposed to the pathogens, the microbes within the vagina. If it comes out C-section, it's basically hopefully within a sterile environment. But from that very moment onwards, they're going to, if you think about a baby that's delivered by C-section versus vaginal delivery is that they are already going to have a different microbiome going forwards, almost through to adulthood, where things tend to balance out a little bit more. If there's different feeding, breast versus formula, okay, are there going to be microbes around the nipples? Yes. And then family, friends, pets, and everything else in the surroundings. That first major burst of exposure to pathogens is during that immediate vaginal birth and then handling. For those of you who have had children, how quickly do you want your hands on them? Yes, that is the beginnings of the establishment of that microbiome. Talk about that. And again, bottle-fed babies are going to be getting different microbes in comparison with breastfed. Okay. There's a difference in the carbohydrates that you get from breast milk versus formula. So therefore, different bacteria will feed on different sugars. So that will affect the microbiome of the gut. You can even have differences in microbiome from child to child in the same household because of these variations. And our microbiomes are continually changing as, you know, I'm about to get a delivery from that sneeze. So this chart, you can just look through it through time. It just goes through colonization, vaginal versus cesarean. With, at least in the early stages, the main exposure to, from a vaginal birth is going to be lactobacillus. And a few other variations. Yeah, we don't really care about these, but just understand that there's going to be a difference from one to the other. Lactobacilli. But with cesarean, it's strange enough, staph. So, gee, during this relatively early stage, you've already got a significant difference in microbiome, and then contact with the outside world going forwards, nursing versus formula. When they're introduced to solid food, all these different things that tie into it. So let's again, this is just using terms that we've conquered before, and it's just clarifying many of them. Pathogenicity, with this one, kind of come to this anyway, the ability of a microorganism to get in and cause disease, physical and physiologic changes to a host, which can lead to changes to health, possible disease, possible death. So when we talk about a primary pathogen, these ones can knock down a perfectly healthy host. Can be perfectly healthy, going to the gym, eating well, hydrating, everything else, and then out of nowhere, you get the flu. That would be a primary. No matter what, this thing's going to get you at some point. Okay? There's a difference here. Opportunistic pathogens, they're already on you. They are in a symbiotic relationship with their surroundings. But if something changes, like you're already fighting off a primary pathogen, opportunistic pathogens can then suddenly decide to run rampant. An example, it doesn't mean to be one bacteria kicking in because of another bacteria. This can be, if you remember the Spanish flu, what was killing most people? It was bacterial pneumonia, because these individuals have already had their pulmonary systems blasted by flu A, and then this bacterial infection that finished them off. The bacterial infection was already there, it was an opportunistic infection. When we talk about virulence, we already talk about this pili, toxins, how fast they replicate. Okay? Obviously, something that is highly transmissible, highly able to get from one person to another, able to cause significant disease, highly virulent. Flu can be highly virulent. Influenza A, again, as an example, highly virulent. Flu C, not so much. lots of factors involved. But with that, we don't just take into consideration what's happening with the pathogen itself. What will influence the virulence of the pathogen is the host it's trying to get into. You have a young, fit athlete, again, like some of you in this room used to be. Right there. Keep that in there. I was there once. Anyway, so 4 minutes 30 for a mile. Come on, now this.
Oh, 4:30.
I actually ran a mile at 4:30.
Wow.
Once.
Under a mile.
It was actually a mile, and it wasn't directly downhill, and it wasn't vertical. It wasn't falling. Oh, thank you. It is slow this morning. So, I'll get this up. Oh, I've got a retort that I'm not going to use because it's a bit nasty. So just the axis.
Lost
Meanwhile, Americans say internet.
What's that?
I said, meanwhile, Americans say words like internet.
Internet, yeah. Oh, the internet. I'm sorry, I had more letters in there. Okay, anyway, back onto this before we start getting off track. Okay, so the virulence of the pathogen is based. The virulence of the pathogen isn't just based on the pathogen itself, but on the host it's trying to get into. If a pathogen is lacking the ability to get into a particular host, it's avirulent, it doesn't get in. Does it mean that's going to be avirulent to all hosts? No, because these things will have to different species, so that will be species dependent. This is going to be stuff that should be nice and simple. The problem is that when I've done this before, I've had people not know what these mean, or at least understand the concepts. What's a symptom? It's a subjective finding. How many findings are you going to have that's subjective? You do an orthopedic test on a patient and they say it hurts. What's that? That's actually a subjective finding. You elicit it, but the patient's response is subjective. Now you say, is that pain out of 10? Where is that pain? Now you're getting the science application of that. So there's a little bit of wriggle room. So it's science observable and objective, hopefully repeatable findings. If you do a straight leg raise in a patient, you should get roughly the same results each time. Of course, if they've got an inflamed sciatic nerve and you repeatedly do a straight leg raise, what do you think is going to happen to them after a while? Yeah, not so much range of motion. So when we get to diagnosis, we are making an educated, hopefully educated guesstimate about what's actually going on with that patient. How often do they get emergency room diagnoses incorrect? So doing it on the fly, 50% of the time roughly. How often are you going to get diagnoses wrong in your office? Depending on the doc, 10 to 20% of the time. So what do we do in those situations? Again, it's a guessing game. Someone comes in with fatigue and a low back pain. What the heck could that be? Anyone want a wild stab in the dark what that could be? It could be they worked out a little bit at the gym yesterday, a little too much. Or they could have pyelonephritis. Or they could be referred pain from the liver and they have Hep B. Yes, there's a lot of guesses that you've got to make going forward. So you do your best. You go through a trial of care. If the patient responds, yay, the diagnosis is probably correct. What happens if the patient doesn't respond to care? You try something new, you dig deeper. Okay? So, again, if a patient is asymptomatic, can you still treat them? Yes? As long as it's not Medicare. Medicare, because they need the ART. Okay? So asymptomatic patient may not have symptoms, but they can still have subjective evidence of clinical disease going on in them. Okay? I just want you may recognize it. If something, the term subclinical, what does that mean? It's just below the horizon. It's bubbling. Okay? Have I seen subclinical patients? Yeah. And then syndrome is just a collection of different signs and symptoms. The term syndrome used to be we have no idea what's going on, so we'll just group it together. Now, are there plenty of diseases that we still use the word syndrome for, but we know the cause? Yeah. It's just we've done research and we've worked out what is actually going on. Okay. So when it comes to pathogens getting into us, there are many different things that can happen. We've got to get the microorganism from where it is to where it needs to be. It has to get from a source to a host. It has to get from a reservoir of infection over there. There has to be some form of transmission. It could be fecal oral, it could be aerosol, it could be fomite, it could be vector. There's a whole bunch of them out there. Even then, when the microbe gets to the host, it has to be able to get into the host. Once it's into the host, it still has to get into target tissue, that tissue specificity we talked about. And when it gets to a target, it has to have a minimum dose to be able to get in there. Okay, we talked about some of these pathogens having an infectious dose. Some of them are in the thousands, tens of thousands, hundreds of thousands. Some of them is like 10, 20, and that's enough to get in and cause disease. As soon as it's there inside the target tissues or the target cells, then they can start the replication process. Congratulations if you're infected. When a replication leads to changes in your physiology, your anatomy, that's disease. There's portal of entry, that's how they get in. There's portal of exit, that's how they get out. Typically, portal of entry, portal of exit are the same. Then again, you might think, well, wait a minute, I swallow a pathogen, that's one portal of entry, it's got a different portal of exit. Well, my argument there is still the gastrointestinal tract it's using. So essentially it's the same portal of entry or exit, it's just one end versus the other. Okay? Many pathogens that are pulmonary in nature, they are then going to be passed on to the next person via aerosols. Okay. So that's something to keep in mind. That diagram just shows some of the steps involved with portal of entry, portal of exit, and how they spread on. At this point, I didn't get questions, but as you're quite, I'm going to keep on going through. So we already talked about this different ways of entries. Breaking the skin, mosquito proboscis punching its way through. Contamination of water or food. All these different things, different types of portal entry in an issue. Okay? So just be aware of that. A term that is crop up is parenteral. Okay, that's basically para, next to, or besides, enteral, gut. Basically anything getting into you via anything but the gut. So a mosquito bite, let's go for a tick bite with Lyme disease. That is parenteral because it's getting into your bloodstream directly, it's not getting into your gut. Okay? So E. coli, giving you food poisoning, that's enteral. Staph aureus causing a furuncle, that's parenteral. So we talked about primary pathogens, opportunistic pathogens, and these terms get used interchangeably, and it does cause a little confusion. So let's see if we can remove a little bit of the confusion going forwards. Okay. So primary pathogen can take you down even if you're nice and healthy. Opportunistic pathogen is going to be there and just step out of the wings onto the stage and cause you mischief if your immune system is compromised. When we talk about infections, a primary infection, again, can strike down a healthy individual. H1N1, H5N1, H5N9, and all these other variations out there. Okay, can cause a primary infection. A secondary infection is something that can occur to someone who's already been weakened by a primary infection. Now, this is the difference. With an opportunistic infection, the pathogen's already there. It's already part of your microbiome, and it takes advantage of that situation, that dysbiosis occurs as the primary. But the secondary infection, that can be not just opportunistic pathogen, or it can be another pathogen from outside that just comes along a little bit later, takes advantage of that. That's the difference between a secondary infection and an opportunistic pathogen. Often, an opportunistic pathogen is already there and it takes advantage of what's going on. Secondary infection is something that occurs after the primary and can be something from outside or something from inside. So there's a slight variation there in terminology. Now that the pathogen's got in, what does it need to do? It needs to get to a specific location. What does polio like to go after? What's its first port of call? It's essentially a gut pathogen that then can get into nerves. Remember, the most common form of polio is abortive. It gets in, but it doesn't get into the nerves because it's busy going after your gut. Rabies is nerves. So just be aware that going forwards, if something can get into your bloodstream, it can get anywhere. Just because it can get anywhere doesn't mean it's going to get everywhere. It still has to try and find a host tissue, host cells that it can get into. Some of these pathogens are more fussy than others. Okay. So if you have that bullseye rash in Lyme disease, that's where the tick bite was. That's what's known as an original infection site, so that will be a locus of infection. If someone has a chancre, that is a locus of infection. That's where it starts off. The terms bacteremia, anemia, viremia, azotemia, septicemia, those are the terms that you should have come across before. The difference with, what's the difference between septicemia and sepsis? No. So septicemia is blood poisoning, basically. That is when you have a large amount of bacteria in your bloodstream. Now, what will happen to your immune system in those situations? It's going to go mental. Anyone heard of a cytokine storm? We will later on.
Yeah.
Yeah. So your immune system can go into overdrive trying to fight off pathogens, and ironically, it is the immune response that eventually kills you. So with sepsis, you're having extreme response to pathogens, and your immune system kicks in too effectively, and you start losing your own cells in the process. Liver, kidneys, everything else starts shutting down. So not fun. I do it for time. Okay, so two more of these. So septicemia, what are the biggies? Aureus, lots of gram-positives. Pneumonia, Pogenies, Staph aureus, Staph Pogenies, those are the ones we kind of taught. Septicemia can lead to sepsis. What's it, toxic shock syndrome, those other lovely things. Yeah, these are the two B's. Pneumonia can do that. Enterococcus can do that. E. coli, Pseudomonas. These are big names that we've already covered. You can lead it in that situation can cause problems. So let's be aware going forward. So I don't want you to memorize these, but just be aware of the biggies. We already talked about toxic shock syndrome when it comes to strep, staph, and everything else, so you should be able to make those connections going forward. Last little thing, and then send you on your way. Valence factors. With these, they can be structural or chemical. Structural, there are also environmental ones outside, but when we talk about structural and chemical, we're talking about what the pathogen itself does. Structural, pili, glycocalyx, flagella, structures on the pathogen itself that help it become, say, motile or hold on. When your immune system is trying to get rid of it, you're trying to stop your skin off, and yet the stuff is still holding on. Biofilms are part of that. The capsules themselves on some of these pathogens will allow them to survive the acid bath of the stomach, getting into your small intestine. Lots of lovely intestinal parasites will do that. It's almost like they know that they're going to be swimming in acid for a little bit, but then they are into your small intestine and then they start going through the usual processes. We have certain pathogens out there that will use our immune cells to their own advantage. Epstein-Barr loves to get into what type of cell? B cells. Do you think that might mess up your ability to fight against Epstein-Barr if it's already behind your defenses? HIV does the same thing. It's the macrophages that transfer HIV from the area of insult, the injury, the port of entry, over to your T cells, or help T cells. We'll talk about those next week. Eli, yes, we know about those. We know about surface proteins the same way. We know about spikes on viruses. If something is able to glue itself in place and cover it with itself with a biofilm or has got a calyx, it's hard to get rid of. Type 4 Eli, sorry, involved motion, flagella, so these things are motile, not just mobile. They will run towards targets and run away from enemies. Some pathogens will just adhere to the surface area and they'll release toxins, and it's the toxins that are going to change the environment within the host to suit the pathogen. That's not just, these things will release toxins to change the environment, but they can also release enzymes to dissolve the surrounding cells to release food. And that just shows them sticking like that and how they can then join together to create a biofilm. What a biofilm just to protect this environment. It's been over and over. My apologies. Okay, that's it. We'll stop there.